Animal breeding refers to the controlled propagation of domesticated animals to improve desirable traits such as milk production, meat quality, egg laying capacity, disease resistance, fertility, and growth rate. It forms a critical pillar in animal husbandry and sustainable livestock development. In the context of CSIRNet JRF Life Sciences, questions focus on classical and modern genetic principles applied to animal breeding, inheritance of quantitative traits, artificial selection strategies, and integration of molecular tools like marker-assisted selection and genomic selection. The foundation of animal breeding lies in population genetics and quantitative genetics. Traits of economic importance in animals are usually polygenic, influenced by multiple genes, and show continuous variation, making quantitative trait loci QTL mapping and heritability estimation essential components of animal breeding programs. Selection intensity and response to selection depend on the heritability of traits and the selection differential, as explained by the Breeder's equation, R equals H squared times S, where R is the response, H squared is heritability, and S is selection differential. Inbreeding and outbreeding are two principal strategies in classical breeding. Inbreeding involves mating between related individuals to concentrate desirable alleles but increases the risk of inbreeding depression, where deleterious recessive alleles become homozygous. This is sometimes strategically used to fix desirable traits in pure lines, such as in laboratory model organisms like mice. Outbreeding, including crossbreeding, outcrossing, and species hybridization, aims to enhance heterozygosity and exploit heterosis, hybrid vigor, leading to improved performance in F1 individuals. For example, crossbreeding of Bos indicus, Indian breeds with Bos taurus, exotic breeds, has enhanced milk productivity in cattle under Indian climatic conditions. Artificial insemination (AI) is a pivotal tool in modern animal breeding, allowing for genetic dissemination from elite males across large populations, even internationally. It reduces disease transmission and improves selection accuracy. AI forms the backbone of progeny testing, wherein the performance of offspring is evaluated to determine the genetic merit of breeding sires. This is a common practice in dairy cattle improvement programs. Open nucleus breeding systems (ONBS) represent structured breeding strategies where superior animals are maintained in a nucleus herd, and genetic gains are disseminated to commercial herds. This hierarchical model helps maintain genetic progress and control in large-scale programs. The integration of molecular markers in animal breeding has revolutionized selection accuracy. Marker-assisted selection (MAS) uses DNA markers, e.g., SNPs, microsatellites tightly linked to QTLs or major genes. For instance, selection for disease resistance in poultry against Marek's disease or improved litter size in pigs is facilitated by MAS. However, many traits are influenced by numerous loci with small effects, requiring genomic selection strategies. Genomic selection (GS) involves whole genome SNP profiling and statistical modeling to estimate genomic estimated breeding values (GEVs) without waiting for phenotypic data. It has reduced generation intervals and increased accuracy, especially in cattle and pigs. Genome-wide association studies (GWAS) identify genomic regions associated with traits, enabling finer genetic dissection of complex traits. Imputation techniques help reduce genotyping costs by using reference panels to predict untyped genotypes in breeding populations. Reproductive technologies like embryo transfer, in vitro fertilization (IVF), and cloning, e.g., somatic cell nuclear transfer (SCNT), are used to propagate elite genotypes. Cloning allows duplication of high-yielding animals, but ethical concerns, reduced lifespan, and abnormal development limit its commercial application. With the advent of CRISPR, SOS9, and other gene editing tools, animal breeding is now entering a precision era. Targeted genome editing has created disease-resistant pigs, e.g., PRRS resistant, and polled cattle hornless, avoiding dehorning. These technologies promise rapid genetic improvement but raise regulatory, ethical, and consumer acceptance issues. In India, government schemes like National Artificial Insemination Program (NAIP) and Rashtriya Gokul Mission aim to improve indigenous breeds and promote sustainable livestock production. Regulatory frameworks such as breed societies, performance recording systems, and genetic evaluation reports are part of structured breeding programs. In conclusion, animal breeding integrates classical genetic approaches, reproductive technologies, and cutting-edge genomics to improve animal productivity, health, and welfare. For CSIRNet JRF aspirants, mastering these concepts, including genetic gain, selection indices, inbreeding coefficient, heritability estimation, and applications of MAS and GS, is critical to tackle applied biology questions effectively. Previous year CSIRNet questions with answers. CSIRNet December 2015. What is the main advantage of genomic selection over marker-assisted selection in animal breeding? A genomic selection uses fewer markers. B genomic selection does not require genotyping. C genomic selection predicts breeding value using genome-wide markers. D genomic selection is less accurate than MAS answer. C genomic selection predicts breeding value using genome-wide markers. CSIRNet June 2017. In animal breeding programs, which of the following strategies is commonly used to reduce the risk of inbreeding depression? A. Artificial insemination with elite sires only. B. Inbreeding within small isolated populations. C. Crossbreeding between unrelated breeds. D. Selection based on phenotype only answer. C. Crossbreeding between unrelated breeds. CSIRNet Deck 2019. Which of the following statements regarding marker-assisted selection (MAS) in livestock is true? A. MAS is only useful for monogenic traits. B. MAS cannot be used for traits with low heritability. C. MAS enables early selection before trait expression. D. MAS increases phenotypic variability answer. C. MAS enables early selection before trait expression. Let me know the next topic, or if you'd like to proceed to another unit in the CSIRNet Life Sciences syllabus. You said. Including marker-assisted selection. ChatGPT said. Here's the extended CSIRNet JRF Life Sciences lecture on breeding in animals, including marker-assisted selection, maximum word limit, paragraph format, highly exam-focused, with previous year questions. Animal breeding is the science of genetically improving livestock by selecting individuals with superior traits to produce offspring with enhanced performance. It combines traditional methods like selective mating and crossbreeding with modern molecular tools. The fundamental goal is to improve economic traits such as growth rate, disease resistance, reproductive efficiency, and product quality milk, meat, eggs, wool, etc. In the CSIRNet Life Sciences context, this topic merges quantitative genetics with biotechnology, making it a high-yield area for applied biology questions. Classically, animal breeding relied on phenotypic selection and pedigree-based selection, where breeders identified superior animals based on observable characteristics or family history. However, this approach has limitations, especially for traits with low heritability or those expressed late in life, e.g., fertility or longevity. To overcome this, estimated breeding values (EBVs) are calculated using statistical models like LUP, best linear unbiased prediction incorporating phenotypic, pedigree, and environmental data to predict genetic merit. These values are used in progeny testing, especially in dairy cattle, where the genetic worth of sires is inferred from the performance of their daughters. The introduction of molecular markers marked a paradigm shift in breeding strategies. A molecular marker is a DNA sequence associated with a trait of interest and can be used to track the inheritance of that trait across generations. In marker-assisted selection, MAS breeders use DNA markers linked to quantitative trait loci (QTLs) to select animals carrying favorable alleles, even before the traits are expressed. This is especially useful for traits difficult to measure or those with low heritability, like disease resistance or fertility. MAS typically involves three main steps: 1. Identification of molecular markers associated with a trait, e.g., SNPs, SSRs. 2. Genotyping of animals for those markers, and 3. Selection of individuals carrying desired alleles. For instance, in pigs, markers linked to the RYR1 gene help eliminate the allele responsible for porcine stress syndrome. In cattle, markers linked to DGAT1 gene are used for improved milk fat content. MAS reduces generation intervals and increases the precision of selection, especially when phenotypic data is unavailable or expensive to obtain. While MAS is effective for oligogenic traits controlled by a few major genes, its power is limited for polygenic traits, where the genetic basis is spread across many loci. To overcome this, genomic selection (GS) is employed. GS uses genome-wide SNP data to estimate genomic estimated breeding values (GEVs) using statistical models like GBLUP or Bayesian approaches. Unlike MAS, GS captures the cumulative effect of thousands of loci, making it superior for traits like milk yield, carcass quality, or disease resistance. Examples of MAS in animal breeding include selection for scrappy resistance in sheep using the PRNP gene marker. Selection against bovine leukocyte adhesion deficiency (BLAD) in cattle using DNA testing. Use of PCB gene marker in sheep to improve fecundity. These programs have demonstrated increased accuracy, reduced costs, and shortened selection cycles. Breeding strategies also include inbreeding, line breeding, and crossbreeding. While inbreeding increases homozygosity and can fix desirable traits, it risks inbreeding depression, which reduces fitness. Hence, controlled outcrossing is often used to maintain genetic diversity. Crossbreeding combines breeds with complementary traits, often exploiting heterosis, hybrid vigor, which enhances traits like fertility, survivability, and growth rate in the F1 generation. Modern biotechnology has further enriched breeding programs. Artificial insemination (AI) facilitates widespread use of genetically superior males. Embryo transfer technology (ETT) and in vitro fertilization (IVF) accelerate multiplication of elite females. Somatic cell nuclear transfer (SCNT) or cloning allows exact replication of elite genotypes. However, cloning suffers from ethical concerns, low success rates, and developmental anomalies. CRISPR Cas9 gene editing now offers precise manipulation of the animal genome. It enables the insertion, deletion, or replacement of specific DNA sequences, allowing the creation of transgenic or gene-edited animals with improved traits. For example, pigs resistant to PRRS virus have been developed using CRISPR. Similarly, cold cattle have been engineered to eliminate the need for horning. For sustainable animal breeding, it's essential to balance genetic gain with conservation of biodiversity. Indigenous breeds often possess superior adaptability, disease resistance, and lower input requirements. Programs like Rashtriya Gokul Mission in India aim to conserve and improve native breeds using genomic and reproductive technologies. In conclusion, animal breeding today is a sophisticated interplay of classical selection, quantitative genetics, reproductive technologies, and molecular genomics. CSIRNet aspirants must understand the principles of heritability, selection response, QTLs, MOS, GS, and gene editing tools to tackle applied questions effectively. Previous year CSIRNet questions with answers. CSIRNet Deck 2016. Which of the following best explains the advantage of marker-assisted selection over traditional phenotypic selection? A MOS is only effective for traits with high heritability. BMOS requires no knowledge of gene function. CMOS allows selection before trait expression. DMOS works only in plants. Answer. CMOS allows selection before trait expression. CSIRNet June 2018. In animal breeding, which strategy is most suitable for improvement of traits with low heritability? A phenotypic selection. B crossbreeding. C marker-assisted selection. D random mating. Answer. C marker-assisted selection. CSIRNet Deck 2022. Which gene is used as a marker in MOS to improve meat quality in pigs? A PCB. BPRNP circa RYR1 DDGAT1 answer. CRYR1. Let me know the next topic or if you want me to continue with the plant breeding lecture including MOS. You said. Marker assisted selection. ChatGPT said. Here's the CSIRNet JRF Life Sciences high level lecture on marker assisted selection MAS. Full length, highly exam focused, structured as a single paragraph, includes previous year questions with answers and years. Marker assisted selection MOS is a powerful technique in modern genetics and breeding that uses molecular markers to identify individuals carrying desirable traits for selection and propagation. Unlike conventional breeding, which relies solely on phenotypic traits, MOS allows the selection of traits at the DNA level even before they are expressed. This is particularly valuable for traits with low heritability, traits expressed late in life like reproductive performance or yield, or those that are difficult, expensive, or destructive to measure, e.g., disease resistance, fiber quality. MOS involves the use of molecular markers such as SSRs, simple sequence repeats, SNPs, single nucleotide polymorphisms, RFLPs, restriction fragment link polymorphisms, and AFLPs, amplified fragment link polymorphisms that are tightly linked to genes or quantitative trait loci, QTLs, controlling traits of interest. The process begins with QTL mapping, where markers closely linked to desired traits are identified using linkage mapping or association studies in a population with known phenotypes. Once these markers are validated, they are used for genotyping individuals in breeding populations. Individuals carrying favorable alleles are then selected, thereby increasing the efficiency and accuracy of breeding programs. MAS has been extensively applied in both plant and animal breeding. In plants, it is used to incorporate traits such as disease resistance, e.g., bacterial blight in rice via EXA21 gene, drought tolerance, yield components, and nutritional improvements, e.g., high fruit content in maize via CRTRB1 gene. In animals, it helps in selecting against genetic disorders, e.g., blood in cattle, or enhancing traits like growth rate or carcass quality. The key advantages of MOS are that it reduces the breeding cycle duration, increases selection efficiency, and facilitates gene pyramiding, where multiple favorable genes for the same or different traits are stacked into a single genotype. Unlike phenotypic selection, MOS is unaffected by environmental conditions and can be applied to seedlings or juvenile stages, which significantly accelerates the process. One limitation of MOS, however, is its reduced efficacy for polygenic traits, which are influenced by multiple genes with small effects. This has led to the development of genomic selection (GS), which uses genome-wide marker data to predict genetic merit through statistical models, especially useful in traits with a complex genetic basis. In India, several breeding programs have successfully integrated MOS, such as the development of improved Kusavasmati1 with resistance to bacterial blight and Swarna-sub1 rice submergence tolerance. For success in MOS, it is critical that markers be tightly linked to the gene QTL, ideally within 5 cm, have high polymorphism, and be reproducible across generations and genetic backgrounds. Thus, MOS represents a marriage of classical breeding with molecular biology, offering a precise, rapid, and cost-effective route for genetic improvement in agriculture and animal husbandry, and is a frequently tested concept in CSIRNet due to its strong conceptual and applied significance. Previous year CSIRNet questions with answers. CSIRNet Deck 2016. Which of the following best explains the advantage of marker-assisted selection over traditional phenotypic selection? A MOS is only effective for traits with high heritability. BMOS requires no knowledge of gene function. CMOS allows selection before trait expression. DMOS works only in plants. Answer. CMOS allows selection before trait expression. CSIRNet June 2018. In a rice breeding program, a line with resistance to bacterial blight is crossed with a high yielding susceptible line. To introgress the resistance trait, which method is ideal to identify progenies carrying the resistance gene? A backcrossing followed by phenotypic selection. B hybridization without selection. C marker-assisted selection using gene-specific markers. D pure line selection. Answer. C marker-assisted selection using gene-specific markers. CSIRNet Deck 2022. Which of the following is not an advantage of marker-assisted selection? A. It allows gene pyramiding. B. It accelerates breeding cycle. C. It is independent of environmental conditions. D. It always performs better than phenotypic selection in polygenic traits. Answer. D. It always performs better than phenotypic selection in polygenic traits. Let me know the next topic you'd like to continue with. You said. Bioremediation. ChatGPT said. Here is your CSIRNet JRF Life Sciences high quality, full-length lecture on bioremediation. Highly exam-oriented, detailed from base to advanced level, no tables, paragraph format, includes previous year questions and answers with years. Bioremediation is the process of using living organisms, primarily microbes and plants, to degrade, detoxify, or remove environmental contaminants such as heavy metals, hydrocarbons, pesticides, solvents, and industrial waste from soil, water, and air. It is a sustainable and eco-friendly alternative to chemical or physical methods of pollution control and is a critical topic under applied biology for the CSIRNet Life Sciences syllabus. The organisms used for bioremediation, including bacteria, e.g., Pseudomonas, Bacillus, Deinococcus fungi, e.g., Aspergillus, Phanerochaete algae, and even genetically engineered microbes, possess metabolic capabilities to utilize pollutants as energy or carbon sources, or convert them into less harmful forms through enzymatic activity. Bioremediation processes can be classified based on environment into in situ, on-site, and ex situ, off-site techniques. In situ methods like bioimaging, biosparging, and natural attenuation are less disruptive and cost-effective, while ex situ techniques like bioreactors or land farming involve excavation and treatment elsewhere but offer greater control. A key mechanism in microbial bioremediation is metabolic transformation, where microbes enzymatically break down organic pollutants. For example, Pseudomonas bacteria degrades toluene and xylene through dioxygenase pathways. In co-metabolism, microbes degrade contaminants incidentally while metabolizing another compound. This is common in the degradation of trichloroethylene (TCE) by methane oxidizing bacteria. Bioaugmentation involves introducing specific strains of microorganisms with desired degradation capabilities into contaminated environments to enhance remediation. In contrast, biostimulation adds nutrients or electron acceptors like nitrate, phosphate, or oxygen to stimulate the native microbial community. Heavy metal bioremediation often involves bioabsorption, where metals like lead, mercury, cadmium, or chromium bind to the microbial cell surface through functional groups, e.g., carboxyl, amino, phosphate, or bioaccumulation, where they are transported into the cell. Some bacteria also chemically reduce toxic metals. For example, Geobacter sulfurregisens reduces soluble UB to insoluble UIB. Phytoremediation, another form of bioremediation, uses plants such as Brassica umbia, Helianthus annuus, or Populus species to absorb, accumulate, and degrade pollutants. Its types include phytoextraction, metal uptake, phytodegradation, enzymatic breakdown, phytostabilization, immobilization of contaminants, and rhizofiltration, root-based absorption from water. Modern bioremediation employs genetically engineered microorganisms, GEMs, to enhance pollutant degradation. One classic example is Superbug, Pseudomonas developed by Dr. Ananda Chakrabarty, capable of degrading multiple hydrocarbons in oil spills. Another is the engineering of E. coli to express meroperon, enabling detoxification of mercury. Use of GEMS in the field requires strict regulatory monitoring due to potential biosafety and ecological concerns. Recent advancements include the use of synthetic biology, metagenomics, and nanobiotechnology to design microbial consortia or enzyme systems that are tailored for specific contaminants and resistant to harsh conditions. Bioremediation is influenced by environmental parameters like pH, temperature, oxygen availability, salinity, and the bioavailability of the contaminant. Contaminants embedded in non-aqueous phase liquids, NaPLs, or bound to sediments are often poorly bioavailable, limiting biodegradation. Surfactants, either synthetic or biosurfactants produced by microbes, are sometimes used to enhance contaminant solubilization. Monitoring bioremediation progress involves assessing microbial population dynamics, contaminant disappearance, metabolic byproducts, and gene expression profiles using molecular tools such as qPCR, metagenomics, and fluorescent probes. Bioremediation has been successfully applied in many real-world contexts: oil spills, e.g., Exxon Valdez pesticide cleanup, e.g., organochlorines, industrial effluent treatment, e.g., textile dyes, and restoration of heavy metal contaminated mining sites. Despite its effectiveness, bioremediation faces challenges such as incomplete degradation, accumulation of toxic intermediates, and the slow rate of microbial action under extreme environmental conditions. Regulatory and public acceptance also affect its implementation. However, with ongoing innovations in microbial engineering, systems biology, and environmental monitoring, bioremediation remains a cornerstone of sustainable pollution management and a frequently tested concept in CSIRNet. Previous year CSIRNet questions with answers. CSIRNet Deck 2017. Which of the following microbial genera is commonly associated with degradation of aromatic hydrocarbons in polluted environments? A. Rhizobium. B. Clostridium. C. Pseudomonas. D. Streptococcus. Answer: C. Pseudomonas. CSIRNet June 2020. Phytoremediation of heavy metals primarily involves which of the following mechanisms? A. Hydrolysis of pollutants. B. Uptake and accumulation in plant tissues. C. Bacterial fermentation and rhizosphere deevaporation through leaf stomata. Answer: B. Uptake and accumulation in plant tissues. CSIRNet Deck 2019. The function of biosurfactants in bioremediation is to: a. Kill non-degrading bacteria. B. Chelate heavy metals. C. Increase pollutant solubility and bioavailability. D. Enhance plant root growth. Answer: C. Increase pollutant solubility and bioavailability. Let me know the next topic you'd like me to cover with the same level of detail. You said, phytoremediation. ChatGPT said. Here is your CSIRNet JRF Life Sciences in-depth, high-standard lecture on phytoremediation. Extensive, exam-focused, no tables, structured in paragraph form, includes previous year CSIRNet questions with answers in years. Phytoremediation is a cost-effective, environment-friendly biotechnological approach that uses green plants and their associated microbiota to detoxify, extract, immobilize, or degrade pollutants from soil, water, and air. It is a subset of bioremediation and is particularly effective for removing heavy metals, radionuclides, pesticides, petroleum hydrocarbons, and excessive nutrients from contaminated. Subset of bioremediation is particularly effective for removing heavy metals, radionuclides, pesticides, petroleum hydrocarbons, and excessive nutrients from contaminated sites. This technique leverages natural plant processes like uptake, metabolism, transpiration, and root microbe interactions to mitigate contamination, and has emerged as a sustainable alternative to physical and chemical remediation techniques. The key to successful phytoremediation lies in the selection of appropriate plant species, often referred to as hyperaccumulators, which are capable of tolerating and accumulating large amounts of specific contaminants without phytotoxic effects. Examples include Brassica euphia, Indian mustard, for lead and chromium, Helianthus annuus, sunflower, for uranium, Thalassophila caryolescens for zinc and cadmium, and P. terra spitata for arsenic. Phytoremediation comprises several strategies based on the type of contaminant and its fate in the plant. Phytoextraction, phytoaccumulation, is the uptake and concentration of contaminants, usually heavy metals, in the harvestable parts of plants. The success of this strategy depends on the plant's biomass and its ability to translocate metals from roots to shoots. After harvesting, the biomass can be incinerated or processed to recover valuable metals. Phytomining. Phytostabilization involves the immobilization of contaminants in the root zone, reducing their bioavailability and leaching into groundwater. This method is ideal for preventing the spread of toxic metals like cadmium, zinc, or lead in mine tailings or industrial waste sites. Phytodegradation, phytotransformation, refers to the uptake and enzymatic breakdown of organic pollutants such as herbicides, pesticides, explosives, and solvents within plant tissues using enzymes like peroxidases, lacases, or nitroreductases. Plants like Populus species and Cannabis sativa are effective in degrading organic contaminants. Phytovolatilization involves the uptake of volatile contaminants like mercury or selenium and their release into the atmosphere in a less harmful form. While this technique reduces soil or water contamination, it transfers the pollutant to the air and is therefore controversial for highly toxic elements. Rhizofiltration, another common method, uses plant roots to absorb, concentrate, and precipitate metals from aqueous waste, typically employed for wastewater or groundwater cleanup. Plants like Lindodium, Eichhornia, and Beteria are used in such systems. The success of phytoremediation depends on factors such as plant root depth, growth rate, tolerance to pollutants, and rhizospheric interactions. The rhizosphere, the soil region influenced by root secretions, plays a crucial role as it hosts microbial populations that contribute to degradation, mobilization, or immobilization of contaminants. Plant microbe consortia, including nitrogen-fixing bacteria, mycorrhizae, and endophytes, can significantly enhance phytoremediation efficiency. For instance, arbuscular mycorrhizal fungi improve plant growth under metal stress and increase uptake of phosphorus and micronutrients. Phytoremediation can be further enhanced by genetic engineering, where plants are modified to express metal-binding proteins like metallothioneins, phytochelatins, or foreign enzymes capable of breaking down organic toxins. Transgenic Arabidopsis expressing bacterial mercuric reductase (MERA) and organomercurial merged genes have shown enhanced mercury volatilization. Despite its potential, phytoremediation has limitations. It is slower than conventional methods, often restricted to surface contamination, and is ineffective in highly contaminated or toxic soils where plant survival is compromised. However, with increasing pollution from mining, industry, and agriculture, phytoremediation is gaining recognition for its ecological sustainability and low operational costs. It also has aesthetic and land restoration value and is being incorporated into green technologies for urban landscaping, constructed wetlands, and biomining. In India, phytoremediation is being explored for cleaning fly ash dumps, tannery effluent zones, and arsenic-contaminated regions in West Bengal and Assam. The method is a regular topic in CSIRNet Life Sciences due to its integration of molecular biology, environmental science, and plant biotechnology, making it essential for applied research and real-world environmental management. Previous year CSIRNet questions with answers. CSIRNet June 2019. Which of the following plant species is known for its capacity to hyperaccumulate arsenic from soil? A. Brassica euphia. B. P. terra spitata. C. Arabidopsis thaliana. D. Oryza sativa. Answer: B. P. terra spitata. CSIRNet Deck 2016. What is the primary mechanism in phytostabilization? A volatilization of contaminants through stomata. B. Accumulation of metals in shoots. C. Degradation of pollutants inside the leaf cells. D. Immobilization of contaminants in the rhizosphere. Answer: D. Immobilization of contaminants in the rhizosphere. CSIRNet Deck 2020. Which of the following is not typically considered a phytoremediation strategy? A. Phytomining. B. Rhizofiltration. C. Phytodegradation. D. Biosparging. Answer: D. Biosparging. Let me know the next topic you'd like me to cover with the same depth and structure. You said, biosensors. ChatGPT said. Here is your CSIRNet JRF Life Sciences High Standard, detailed lecture on biosensors. CSIRNet focused, paragraph format, maximum word limit, no tables, includes previous year questions with answers in year. Biosensors are analytical devices that combine a biological recognition element with a physiochemical transducer to detect and quantify analytes with high specificity and sensitivity. These devices are widely used in medical diagnostics, environmental monitoring, food safety, and bioprocess control. A biosensor consists of three primary components. 1. A biorecognition element, such as enzymes, antibodies, nucleic acids, whole cells, or receptors that selectively interact with the target analyte. 2. A transducer that converts the biological interaction into a measurable signal, such as optical, electrochemical, piezoelectric, or thermal. And 3. A signal processing system that amplifies, interprets, and displays the signal. The central principle of biosensors lies in their ability to transduce a biochemical event into a quantifiable output, often in real time, allowing for rapid and on-site detection. Biosensors are classified based on the type of transducer or biological element employed. Electrochemical biosensors are the most commonly used to detect changes in current, amperometric, potentiometric, or impedance conductometric in response to biochemical reactions. These are widely applied in glucose monitoring using glucose oxidase. Optical biosensors use light absorption, fluorescence, or surface plasmon resonance (SPR) to detect interactions such as antigen-antibody binding or DNA hybridization. SPR-based biosensors have become indispensable in studying molecular interactions without the need for labeling. Thermal biosensors, or calorimetric sensors, detect heat changes during enzymatic reactions, while piezoelectric biosensors rely on changes in mass or acoustic waves upon analyte binding, typically using quartz crystal microbalances. The choice of biosensor depends on the nature of the analyte, required sensitivity, and field of application. The biological recognition elements determine the specificity of biosensors. Enzyme-based biosensors catalyze substrate conversion, and the product or associated change, pH, temperature, etc., is measured. For example, urease-based sensors detect urea levels via the pH change produced by ammonia formation. Antibody-based biosensors, or immunosensors, rely on antigen-antibody binding and are used in the detection of disease markers like HIV proteins or cancer antigens. Nucleic acid biosensors detect complementary DNA or RNA sequences, with applications in pathogen detection and gene expression profiling. Aptamer-based biosensors, using short synthetic oligonucleotides, offer high affinity and specificity, especially for small molecules, and are gaining popularity due to their stability and reusability. Whole cell biosensors use metabolically active cells that respond to toxins, pollutants, or metabolic changes. These are used in environmental and toxicological monitoring. Biosensor design and signal amplification have advanced with nanotechnology and synthetic biology. Nanomaterials such as gold nanoparticles, carbon nanotubes, graphene, and quantum dots enhance the sensitivity and stability of biosensors by increasing surface area and electron transfer rates. For example, gold nanoparticle modified electrodes enhance electron transfer in electrochemical biosensors, while quantum dot labeled antibodies improve fluorescence detection. Additionally, synthetic biology enables the construction of genetically engineered microbes or cell-free systems that act as programmable biosensing platforms. These advances have led to the development of lab-on-a-chip devices and point-of-care diagnostics that integrate biosensors into compact, user-friendly systems suitable for clinical and field use. Applications of biosensors span diverse domains. In medical diagnostics, biosensors detect biomarkers for diseases like diabetes, glucose sensors, cancer, tumor markers, cardiac conditions, troponins, and infectious diseases, antigen, antibody detection. In environmental monitoring, biosensors detect pollutants such as heavy metals, pesticides, and pathogens in water and soil. For instance, genetically engineered bacterial biosensors expressing GFP, green fluorescent protein, in response to mercury are used for toxicity screening. In agriculture, biosensors are employed to monitor crop pathogens, soil nutrient levels, and pesticide residues. Food industry applications include detecting contaminants like aflatoxins, foodborne pathogens e.g., salmonella, and spoilage indicators. Industrially, biosensors are used in bioprocess monitoring, ensuring real-time control over fermentation parameters such as glucose, lactate, or oxygen levels. Despite their wide applications, biosensors face challenges including stability, reproducibility, shelf life, and non-specific binding, especially under real-world conditions. Advances in material science, immobilization techniques, and miniaturization are overcoming these limitations. One major trend is the integration of biosensors with smartphones and wearable devices for continuous health monitoring. Glucose sensors embedded in wearable patches or contact lenses are examples of next-generation biosensing technologies. In the context of CSIR net life sciences, biosensors are often linked to applied biology, immunology, enzymology, nanobiotechnology, and environmental biology. Questions test understanding of biosensor types, mechanisms of detection, enzyme antibody-based applications, and current trends in biosensing technology. Mastery of these concepts is essential for both conceptual and application-based questions in the exam. Previous year CS CSIR net focused, paragraph format, maximum word limit, no tables, includes previous year questions with answers in year. Biosensors are analytical devices that combine a biological recognition element with a physiochemical transducer to detect and quantify analytes with high specificity and sensitivity. These devices are widely used in medical diagnostics, environmental monitoring, food safety, and bioprocess control. A biosensor consists of three primary components. 1. A biorecognition element, such as enzymes, antibodies, nucleic acids, whole cells, or receptors that selectively interact with the target analyte. 2. A transducer that converts the biological interaction into a measurable signal, such as optical, electrochemical, piezoelectric, or thermal. And 3. A signal processing system that amplifies, interprets, and displays the signal. The central principle of biosensors lies in their ability to transduce a biochemical event into a quantifiable output, often in real time, allowing for rapid and on-site detection. Biosensors are classified based on the type of transducer or biological element employed. Electrochemical biosensors are the most commonly used to detect changes in current, amperometric, potentiometric, or impedance conductometric in response to biochemical reactions. These are widely applied in glucose monitoring using glucose oxidase. Optical biosensors use light absorption, fluorescence, or surface plasmon resonance (SPR) to detect interactions such as antigen-antibody binding or DNA hybridization. SPR-based biosensors have become indispensable in studying molecular interactions without the need for labeling. Thermal biosensors, or calorimetric sensors, detect heat changes during enzymatic reactions, while piezoelectric biosensors rely on changes in mass or acoustic waves upon analyte binding, typically using quartz crystal microbalances. The choice of biosensor depends on the nature of the analyte, required sensitivity, and field of application. The biological recognition elements determine the specificity of biosensors. Enzyme-based biosensors catalyze substrate conversion, and the product or associated change, pH, temperature, etc., is measured. For example, urease-based sensors detect urea levels via the pH change produced by ammonia formation. Antibody-based biosensors, or immunosensors, rely on antigen-antibody binding and are used in the detection of disease markers like HIV proteins or cancer antigens. Nucleic acid biosensors detect complementary DNA or RNA sequences, with applications in pathogen detection and gene expression profiling. Aptamer-based biosensors, using short synthetic oligonucleotides, offer high affinity and specificity, especially for small molecules, and are gaining popularity due to their stability and reusability. Whole cell biosensors use metabolically active cells that respond to toxins, pollutants, or metabolic changes. These are used in environmental and toxicological monitoring. Biosensor design and signal amplification have advanced with nanotechnology and synthetic biology. Nanomaterials such as gold nanoparticles, carbon nanotubes, graphene, and quantum dots enhance the sensitivity and stability of biosensors by increasing surface area and electron transfer rates. For example, gold nanoparticle modified electrodes enhance electron transfer in electrochemical biosensors, while quantum dot labeled antibodies improve fluorescence detection. Additionally, synthetic biology enables the construction of genetically engineered microbes or cell-free systems that act as programmable biosensing platforms. These advances have led to the development of lab-on-a-chip devices and point-of-care diagnostics that integrate biosensors into compact, user-friendly systems suitable for clinical and field use. Applications of biosensors span diverse domains. In medical diagnostics, biosensors detect biomarkers for diseases like diabetes, glucose sensors, cancer, tumor markers, cardiac conditions, troponins, and infectious diseases, antigen, antibody detection. In environmental monitoring, biosensors detect pollutants such as heavy metals, pesticides, and pathogens in water and soil. For instance, genetically engineered bacterial biosensors expressing GFP, green fluorescent protein, in response to mercury are used for toxicity screening. In agriculture, biosensors are employed to monitor crop pathogens, soil nutrient levels, and pesticide residues. Food industry applications include detecting contaminants like aflatoxins, foodborne pathogens e.g., salmonella, and spoilage indicators. Industrially, biosensors are used in bioprocess monitoring, ensuring real-time control over fermentation parameters such as glucose, lactate, or oxygen levels. Despite their wide applications, biosensors face challenges including stability, reproducibility, shelf life, and non-specific binding, especially under real-world conditions. Advances in material science, immobilization techniques, and miniaturization are overcoming these limitations. One major trend is the integration of biosensors with smartphones and wearable devices for continuous health monitoring. Glucose sensors embedded in wearable patches or contact lenses are examples of next-generation biosensing technologies. In the context of CSIR Net Life Sciences, biosensors are often linked to applied biology, immunology, enzymology, nanobiotechnology, and environmental biology. Questions test understanding of biosensor types, mechanisms of detection, enzyme antibody-based applications, and current trends in biosensing technology. Mastery of these concepts is essential for both conceptual and application-based questions in the exam. Previous year CSIR Net questions with answers. CSIR Net December 2015. Which of the following components of a biosensor is responsible for converting the biological signal into an electrical signal? A. Bioreceptor. B. Transducer. C. Amplifier. D. Processor. Answer. B. Transducer. CSIR Net June 2018. In an amperometric biosensor used for glucose detection, which enzyme is typically used? A. Glucose isomerase. B. Glucose oxidase. C. Hexokinase. D. Invertase. Answer. B. Glucose oxidase. CSIR Net Deck 2020. Which of the following is not a type of biosensor transduction method? A. Optical. B. Thermal. C. Magnetic resonance imaging. D. Piezoelectric. Answer. C. Magnetic resonance imaging. Let me know your next topic for CSIR Net Life Sciences, and I'll continue in the same style with maximum quality and depth.